I mean, if Jonah and William are fine with starting, I guess our group can go first.
Okay, sounds good. Take it away then. I've given you screen sharing, so you should be able to get started. We're looking forward to hearing from you. Thank you.
Okay. Just need to share the right thing. You guys see it?
Yes.
Great. Okay, I'm Jonah. Today we're going to be looking at the role of instabilities and formation of the Galilean satellites of Jupiter. This is a presentation by me, Jonah Goldstein, Anton Perez, William Zhao. Our teacher was Mr. Gardner, and our advisors are Max Goldberg and Dr. Konstantin Batygin. First, as a sort of introduction to the Galilean moons, they were the first astronomical discovery with a telescope by Galileo. So Io is the smallest of the Galilean moons, and it is the most volcanically active body in the solar system. Europa is the second smallest of the group, and the oceans theoretically could be a place where life could form. Ganymede is the largest moon in the entire solar system, and Callisto is the third largest moon in the entire solar system. Today we will look at Io, Europa, and Ganymede because of a sort of unique, at least not particularly unique, but as a pattern known as orbital resonance. So orbital resonance is a self-correcting sort of orbital chain that keeps many objects throughout the universe in a stable and usually digestible number of orbits. As you can see of the orbital periods listed, 1.769 days for Io, 3.538 for Europa, 7.076 for Ganymede. But if you take a closer look, you will notice that Europa is double that of Io, and Ganymede is double that of Europa and quadruple that of Io. And Jupiter's inner three moons are in a 1, 2, 4 resonance chain, in which, so for every time orbit, Io goes around once, Ganymede goes around a quarter of the time, and Europa goes around half the time. And so after 2 Io orbits, Europa has completed 1 orbit, and after 4 Io orbits, Ganymede has completed 1 orbit, and Europa has completed 2 orbits. This is how it currently is now, but if we turn the clock back 4.5 billion years ago, Jupiter and its satellites could have looked much, much different. So here's our hypothesis on the formation of Io, Europa, and Ganymede. So a few weeks ago, a new image of an exoplanet was found. Here, a planet is being formed from a protoplanetary disk, labeled on the right. Though this planet is already formed, there is more mass surrounding it. That's the little dots you can see in a circumplanetary disk. And that mass is enough to make several moons the size of the Galilean moons. And we believe that this is a very similar case to Jupiter in its early stages. So our hypothesis is that dust coalesced into icy satellites, and then those smaller icy satellites collided into the three moons in resonance that we see currently, and then migrated inwards towards Jupiter and formed a resonance. The idea of collisions of smaller icy satellites has not gotten much limelight in other hypotheses surrounding the formation of the Galilean moons, and it's what we attempted to prove through simulations and a program called Jupyter Notebook and Rebound.
The main laws that are the primary laws that describe planetary motion are Kepler's laws and Newton's law of gravitation. Kepler's first law basically states that all orbits are ellipses in which the central object is at one of the foci of the ellipse. There are 2 major properties that define the shape of an ellipse: its semi-major axis and its eccentricity. The semi-major axis is the point on the ellipse that is farthest away from its center. You can see in the middle, or the picture in the middle, That would be the distance or from the center of the cross to the edge of the ellipse. Kepler's second law basically states that as planets get closer to the or the object that they're orbiting, they tend to move slightly faster, and then the farther away that they are, they'll move slower. Kepler's third law gives us a relationship between the period, that is how long it takes a satellite to orbit the central object, and the semi-major axis of the ellipse that the orbit takes. So it basically states that the period squared is proportional to the semi-major axis cubed, and that basically as the semi-major axis increases, the period will also increase, and vice versa. Newton's law of gravitation essentially tells us that the more massive objects are, the greater the force is between them, and the farther away they are, the less the force, or the smaller the force is between them. As you can see, since the force is proportional to the masses of both of the objects, and force is inversely proportional to the square of the distance between the two objects. Next slide, please, John. So with those previous laws, you could construct a series of differential equations that you can use to describe an orbital system. But issues arise when you start, or when you have three particles, or three or more particles. There is no way to get a closed solution to a system where there are three or more bodies interacting with each other gravitationally. What that means is that you can't use a system, or you can't use initial conditions and time to plug into those differential equations and get a deterministic answer. Because of that, you have to use numerical methods to approximate quantities like the position or the velocities of these particles at later points in time. So next slide, please, Joe. Another aspect of orbits, or another key factor that affects orbits is something called migration. Migration is basically the process by which satellites or objects interact with a protoplanetary disk of gas, and their orbits are altered in such a way where they start moving in closer to the central object that they're orbiting. So in the diagram to the right, you can see how this planet is sort of spiraling inwards towards the object that's orbiting. So migration also plays a role in the formation of resonance chains, like the ones that we see with Io, Ganymede, and Europa. So as planets or satellites migrate inwards, they tend to cause other satellites to start migrating inwards too. In the diagram to the left, you can see as this outermost green satellite begins moving closer towards the object it's orbiting, it starts to force the other satellites into migration as well, and that leads to the formation of resonance chains. Another thing to note about orbits is that they are chaotic, which means that small alterations in the initial conditions can cause drastic differences in the outcomes of those systems. So as time goes on, this protoplanetary disk, it eventually goes away, and that causes instabilities to arise, and which creates which allows for things like collisions between satellites to occur. So next slide, please. So to simulate all of this, we used a library called Rebound, which basically allows us to use various numerical methods to approximate the positions or other quantities associated with these orbital systems. Here in the picture below, you can just see it's initializing a few bodies or satellites and at different positions and periods, and that's how we were able to get our results, which William will now show you.
Yeah, now that Anton talked about the methods, I'll be talking about the results of these simulations. So we kind of started with fewer moons to have a more simpler configuration. And already with 3 moons, we could see the effects of migration here as the periods of the moons are decreasing, which due to Kepler's third law applies that they're getting closer and closer to Jupiter. And we also see how migration is eventually getting turned off because the circumplanetary disk is getting blown away by the sun. And eventually we don't even have, the moons are kind of in a stable configuration. They're not moving towards or away from Jupiter. So that's on the left-hand side of the period versus time graph. You can also see how the simulated moons entered stable resonance chains. So on the right-hand side of the graph, we can see the period ratio. So on the blue curve between the innermost and middle moon, we see that it eventually arrives at 1.5 or a 3/2 resonance. And then on the orange curve between the middle moon and the outermost moon, we see that their period ratio arrives at 1.25 or a 5/4 resonance chain. So that kind of verifies our in predictions with 3 moons, how they're going to be arriving at resonance. Furthermore, we could graph the eccentricities of these moons versus their time. And at the beginning, we have this phenomenon. Oh, I think I should define eccentricity, because so if an eccentricity is closer to 0, that means that the orbit of that moon is more circular, while if the eccentricity is closer to 1, that means it's more elliptical. So at the beginning, when there's still the circumplanetary disk, there's something called eccentricity damping, which is basically keeping eccentricities down and the moon's orbits or the satellite's orbits being more circular. But as eccentricity damping is weakening over time, as you can see on the graph here, the eccentricities of these orbits are increasing. So that's again a verification of our predictions. Then we decided to add more moons, and with more moons, our results got more complicated and more diverse. So for example, in this graph here, we saw how adding moons adding more moons could make resonance chains unstable due to there being more gravitational interactions. So as you can see here, with these 5 moons, we're basically at resonance at 20,000 years, and migration has been turned off. But due to these resonance chains eventually become unstable at around 35,000 years, leading to collisions. And instead of there being 5 satellites, there are basically 2 satellites at the end of the simulation. The reason for this is apparent on the right-hand side of the graph, or the right-hand graph, because eccentricity is increasing. Once eccentricity damping is removed, there is more gravitational interactions or closer gravitational interactions between the moons, eventually resulting in instabilities and moon mergers. However, that's not the only possible scenario that could result. We could also have moons migrating inwards, and these once during the migration period, we could have collisions as well. So on the left-hand graph here, again, we have a period to time graph where the pink moon eventually collides with the brown moon, and then eventually the brown moon will collide with the red moon. So instead of having 7 moons, we end up with 5 moons, or with 5 moons. And these are kind of in resonance at the end of the simulation, as shown by the period ratio to time graph on the right-hand side, where the period ratios become mostly stable at the end of the simulation. So we can have moons colliding during the period where they formed unstable resonance chains, or during migration as well. And then finally, it was also possible to form stable resonance chains, even though there are a large number of moons. So in this simulation, we had 6 moons and they were migrating inwards. And then once dampening was turned off, we could see that they form a pretty stable resonance chain. And we also see a phenomenon that once dampening is removed, we kind of see that, or once migration and dampening is removed, right? We see that the moons are moving further apart from each other, and that's a phenomenon we kind of see very frequently with these types of simulations. And then finally, we also ran various similar simulations to try to observe the number of resulting moons. So we had to run some of these similar simulations because of the chaotic nature of orbits. So we basically just had an initial condition. We changed it a little bit and then we tried to record that the number of moons after finishing the simulation. So for example, on the left-hand side, we had initial conditions similar to a period of like 5, 6, 7. So we had 5 moons with 5, 6, 7, 9, and 11 days respectively. And we sometimes added so that the innermost moon had a period of 5.1 days, or the outermost moon had a period of 11.1 days. And we tried to record the results. So on the left-hand side, with moons starting closer to each other, with satellites starting closer to each other, we had moons coalescing into 1, 2, or 3 moons. And on the right-hand side, with satellites further away from each other, they ended up coalescing into 3 or 4 moons. And that showcases that if we have satellites starting closer together, they usually coalesce into a smaller number of moons. So in conclusion, we can model the Jovian system's resonance through collisions of smaller satellites, but there's also a very large variability in what the resulting system produced by migration satellite mergers looks like. And we can model the Jovian system's resonance because we ended up getting a lot of the same resonance chains within our simulations.
Nice, good work, you all. That's fantastic. Got any questions for them from the other group or the other mentors or teachers? Anyone want to chime in with a question? I have one, and this is probably very naive, but in reality, would the moons coalesce into a larger moon, or would they hit each other and explode into lots of pieces? What would actually happen beyond theory? What would typically happen when they collide?
I guess in this situation, we don't really know particular the exact this is exactly what happened. At least we don't know that yet. So it could have just been that the dust coalesced into just 3 planets, and that just happened. But we believe that they coalesced into smaller icy satellites, and they were much further away from Jupiter, and that those eventually collided with each other, and then those formed the moons.
Thank you.
It's a great question, Mitch. It's a great question.
Thanks. How did you guys collaborate across the distance. Did you meet regularly to talk about who's doing which piece of it, or how did that come together? It's been interesting during COVID to see how all of these projects were so successful from via Zoom, basically. So how did it work for your teams?
We basically had regular meetings where we kind of decide on what we all want to kind of collaborate on doing, like which steps and which programs we want to make. And then that's how we kind of moved forward so that everybody was in sync and how we ran those simulations together.
Yeah, and so Max did a lot of like showing them how to, make sure everybody had Python, and then made sure that everybody could run Rebound, and that we could run the updated version of Rebound, which we needed for several of the pieces. And then so that took like a couple of weeks because it's starting from kind of scratch with Python and like running a console that we could run Python on, because Python didn't actually run on the Windows system, not with Rebound. Anyway, it was a lot of complicated stuff, and it was pretty awesome to see these students like rise to the challenge at every point along the way. And I mean, Max is really patient, really giving like exactly what was needed, wait time, suggestions, finding solutions. So it was awesome to see just the teamwork working in the Zoom system. It's definitely different from my other SRC experience where, we were in a lab working with lasers and you literally hook them up, like, if it's working, instead of you plug it in and you get 100,000 errors, I think was what you got, right? One of those times, Jonah. And it's just, I mean, anyway, it was quite the experience.
Thanks, Jonathan. Max, any thoughts on from your perspective?
Yeah, I think they covered it well. We have like a group Slack channel, and so the students were added to that, which I think hopefully was enjoyable for them, and they came to some of the our regular group meetings just to see what that dynamic is usually like. Yeah, I think it was obviously a challenge, but numerical experiments tend to work fairly well remotely, just because you don't have to be in a lab. And I think like William is, not in Southern California, and that's like really it's good that people are able to kind of join from all over, and so I think that ended up being an asset in some ways.
Great. Dr. Batygin, any thoughts on how it went?
I only have one thought, which is that the key to success is Max Goldberg and Jonathan Gardner and amazing students. I didn't do anything. I mean, quite literally, I just had random musings about satellites, and they listened politely and then got back to actual work. So yeah, it was I'm just really impressed and very, very proud of the students, and very proud of Max, who really took on the the actual role of faculty advisor, while I just kind of sat in the back seat, at best, back seat at best, maybe 2 seats behind. So again, everybody, Anton, Jonah, William, you guys did a fantastic job, and this is great.
Thank you. That sounds like a wonderful team when all the parts are working well together. That's terrific. Any other questions before we move to our next group? If not, we'll go ahead. Sarah and Eli, you want to take it away?
Yeah, sure. Our students prepared the presentation themselves, so we're excited to see it too. But yeah, if you guys want to share your screen and yeah.
Can you guys see?
Yes.
All right, cool. Present now. Oh, my bad. Okay, so this is our SRC final presentation by Benjamin Levid, Emily Huang, Christine Moody, and Kevin Madrano.
How do I go? Okay, so for the project, we were told that we would be using a sorry, that was my mom. Okay, so for the project, we were told that we would be using a labeling tool to label existing databases of species that were taken with camera traps placed in Kenya. Doing so would help improve the AI of the camera traps at labeling and identifying the species. One of our first tasks was to learn and memorize 59 different animals that we would encounter in the interface. After we were told to correct a certain amount of images that the AI had labeled. I think it was around like 15 or so. And then by doing so, it would help us to familiarize ourselves with the program and the type of species that we would see. And lastly, we were asked to correct an image of about 200 images that the AI had previously labeled.
Hi. We created flashcards to help us study the animals that would appear on the interface. So what we did was that we had an original list of all of the animals that would appear on the interface, and all of us took turns creating these flashcards. And whenever we needed help, like when we were in the middle of labeling the images, we would always refer back to the flashcards to help us out. So the flashcards were just used as like the helping tool to help us like label all of the animals and stuff like that. And the link to the flashcards are, on the slide. Some of the flashcard examples, well, how we would format them is that we would try to get like the faces of the animals and like the sides, like the special markings of each animal so that they would be able to be recognized easier, because in the images, some of the images were really not, well, they're like taken like either in the dark or with like a limited light. So we would just need the most identifiable like things like shapes of like their ears or like the side of like an antelope. So we had we labeled like a bunch of antelopes, and we could see like we had to differentiate between them, especially between if they are the same species, like the different types of gazelle and also different types of giraffes. And also like on the next slide, it's we're comparing it to like the bat-eared fox and like a jackal. Like it's really hard to see which one is which in the dark. So we had to really look at the fine details in order to label all of the animals and things like that. And also like look at differences between like the zebras. Those are really hard to differentiate between as well. But like plain zebras have like a bigger stripe, Gavi zebra have like a smaller stripe, things like that. And that's how we identified each one and labeled with the help of the flashcards. And yeah.
Okay, so this is the labeling tool, like the steps to it. So it was pretty simple and straightforward. First, you would use the classes search to find the species that was present in the image. You just type in whatever species it is, and then you click on it. And after that, you would hit the little plus to create a box. And then if you needed to, you could just click on the little minus and then hit the box, and it'll just take it off. third thing was to try to be as accurate as possible in boxing the species presented. Lastly, if you think you labeled all the species that were presented, you could just click on the next button, or if you wanted to go back, click on the previous button. We were also provided with a detailed set of instructions for using the labeling tool. If you want, I can show it like at the end of the presentation. It's just like another like slides to help us. So this is just like a couple of images that we had to label. And then this is what we like considered to be easy, because like the cows, elephants, baboon, the goats, camels, and giraffes, those are pretty straightforward. You would be able to tell what it is, how to label it.
And with that, there also came a bunch of harder images. The camera setup in Kenya did not always take the greatest images of these animals. Sometimes they were at night. So just with that, it's kind of hard to tell, you know, if they had a different type of stripe, their body size, shape, all that different type of stuff, if they had horns. And then on top of that, they wouldn't always take pictures, you know, of their face, something like where you could quickly identify the type of animal it was. Sometimes, as you can see, it would just take pictures of like the rear end of the animal. And just from that, you would have to identify the animal. And these are animals that we're not 100% familiar with. So we had to get pretty familiar with these different types of animals via the flashcards. And yeah, if we had any troubles with identifying these images, we would, I don't know, talk to each other via the Slack channel, discuss what we thought the animal was, and then from there, label it. So yeah, throughout this process, I mean, it was pretty enjoyable. I learned a lot about identifying animals and the different types of animals there are. I mean, coming into this, I didn't know like there were different types of like antelopes. I thought it was just like a singular thing. Same with gazelles. I thought it was just one thing. Giraffes, I thought it was just one thing. Zebras. Go on. I could keep on going, like the different examples. So I learned about a lot about different animals. I learned how to identify them, how to differentiate between them. I found it very difficult to differentiate between like the different types of antelopes, as I said earlier. In the process of labeling all of these and annotating them in the interface was, I mean, it wasn't necessarily too hard, but it did take up a lot of time. It was 200, so yeah.
I thought it was a very fun and new experience. I liked how I got to see many different species up close, such as a leopard and a hippopotamus. And then there were definitely some difficulties, such as having to annotate an image with several of the same species, like cows and sheep, and also camels.
So memorizing the different types of species was like pretty easy, because I was just using the flashcards, you know. The labeling process was straightforward. It just took time, you know, because we had to do 200, so that, you know, took time. And then I found it a little hard to tell apart the antelopes, because, you know, some are very similar looking, and then it all depended on like the quality of the picture stuff. And then an issue in the labeling process was the fact that some of the images were repeated. So then that, you know, that made it take longer to label all 200.
The process of labeling all of the animals was pretty easy and straightforward because the interface we were using was pretty nice. So it was easy to use, and if I ever needed help, I would just go back to refer to the flashcards. But it was like really long process of labeling all of the animals because, as like aforementioned, it was like 200 animals. So I would usually like get distracted really easily, and I would go back to it and start labeling again. And I would always go back to the flashcards just to help check my answers. And I would also ask one of the like advisors if like my answers were correct as well. And other people did that in the group as well. We would ask each other hey, what do you guys think this animal looks like? Different things like that. So work together to help label all of these animals.
As far as what we learned how to identify the different animals in Kenya. We learned how diverse that region is. I mean, it's kind of crazy how many different types of animals, how different they look, all the different shapes and different shapes and sizes that they come in just one area. We learned how to operate an annotation interface. That's something very new for me. Never really did anything like that. Learned how an AI builds up its intelligence, how it becomes smarter, how difficult and long that process is. Realized how difficult it is to distinguish the different types of antelopes, along with that, like other species, not necessarily just antelopes, and that a lot of the primary animals in Kenya were cows, impala, zebras, camels, and goats.
That was great. Thank you. Did you each do 200? Were you each assigned 200
Yeah.
animals? Yeah. And so could someone talk about how your 800 animal identifications will feed into making the AI better? So you've given it some correct answers to help it learn, basically. How does that step work?
So like from the jump, like it would identify an image, like it would take a picture, then it would kind of like guess what type of animal it was. Then from there, like we would correct it. And as time went on, as we did more and more images, the AI system would get closer and closer to the correct image. So, it just took a while for it to recognize different features, characteristics until finally, it's not perfect yet, but at the end, we saw how it was pretty close. I felt at least.
Other questions about how it worked and how the process happened and what their goal was? Constantine.
Maybe I didn't catch this, but was the AI, is this like a pre-trained Google AI that already knows or about animals, or were you training it from scratch?
I don't know if they know the answer to that question because it's just a model I gave them. But this is a model that's been pre-trained by Sarina using a bunch of labeled data from Lila.science. So it's camera trap data. And what she did is the research question we're trying to ask is like, how do you build a machine learning model? How do you adapt it efficiently to a new region where the species set is similar to other things you've seen before, but maybe not explicitly represented? So we had models trained from Snapshot Serengeti, which is like in Serengeti National Park in Tanzania. And then we're using those models to provide weak labels and then correcting them for our network of camera traps, which is in Laikipia, Kenya.
So cool.
And so there's like some species that were not well represented before, some species that sort of no longer exist. And these guys are helping build up both some additional training data for our network, but as well as we're going to be looking at using their labels for evaluation as well. So some of it's going to be for evaluation.
That's pretty cool. That's pretty cool. What's the weirdest? Sorry, I'm just like, what's the weirdest animal that you guys saw? And you were like, this is a Pokemon. This is not like, alive.
The camera one time like recognized like a human face. Someone went up to the camera, I think, and like they took a picture of the person. I was like, oh, okay.
Yeah.
And also a few times the picture took like a picture, but when I looked on the screen there was like no animals there, and also a few times the interface decided to label rocks as like as like animals, but I'm not sure if that answers your question of like the most weirdest, but these are like definitely outliers in what I've seen.
I make that mistake as well. I'm no AI. I'm just me, but I sometimes look at a rock and I'm like, I'm not sure I trust this thing to not bite me. So
What was your favorite animal, you guys, after all this time?
The impala. Yeah,
Why?
it looks pretty unique. I like how it kind of looked, and I thought it was pretty easy to identify, so I kind of liked it for that.
Anyone else?
I like eating a bunch of elephants, especially the baby ones, because they were cute.
That's always my favorite too. The baby animals are the best.
I think mine would be birds, Cory bustard. I think I only saw it one time and I thought it was pretty cool. It just looks interesting.
The Cory bustard is the largest bird that can fly. So it's yeah, unlike the ostriches, which cannot fly. Cory bustards are very big and can still fly. And when they do, they're kind of like B-52s or something. It's pretty impressive.
This seems like a very high traffic area. Is it next to a watering hole, or what's the attraction that brings so many animals past the camera?
So it's a network of 100 cameras, and some of them are at magnet sites. So you have watering holes in Salt Lake. Some of them are on a random grid. We definitely see differences in like the number of animal sightings versus like the camera placement, and that's one of the things that we're investigating. Eli and I went and placed all of these cameras ourselves last January, and it was both of our first time in Africa and both of our first time doing field work as computer scientists, and it was pretty crazy.
Are all the cameras still working, or has anyone like stolen
No,
them?
I actually got back from Kenya like 2 days ago. I went back for the first time, and there were about 32 of the cameras had either gone missing or been destroyed. And I made like a map of sort of how that happened. And then we were actually able to fix about 7 of them by zombieing together not broken pieces of the cameras that got broken. But the ones that were broken, the vast majority were crushed, like literally crushed by elephants. There were also some that got chewed by hyenas. And then there was a woodpecker in one particular area that like took a dislike to the IR sensor and like pecked out the IR sensor on 3 cameras. So yeah.
That should be a website just for people to watch what happens when the animals.
The craziest part is, yeah, sometimes you get like the picture. It's like the elephant's like coming, and then that's like the last photo on the SD card, and the camera's just crushed.
So Benjamin, you mentioned Impala was your favorite. How did you tell the difference between the Impala and the Thompson's Gazelle? Because those always seemed so close together. Like in the wild, I could never pick them out. So
For me, like I noticed that I'm colorblind, so like color differentiating is kind of difficult for me. But I always noticed that there was 3 different shades with the Impala. Like their stomach was pretty white. Then there was like a big, I don't know, kind of large stripe that was like a lighter shade, and then at the end, like most of them would be like brownish. So I mean that's how I'd see it. Thompson doesn't have that, so that's how I recognize that.
Thompsons have a big black stripe on their side too. So that's another.
Oh yeah, I forgot.
And they're smaller, but that's harder to tell if you're going sort of between cameras. So one thing that ecologists do, because most ecologists label all this data by hand. You guys, 200 images is rough. I agree. That's one of the reasons we're trying to build machine learning models for this. But ecology grad students who work with camera traps, it's usually on the order of like 250,000 images that they have to label. So like it gets rough. Yeah, if you just look at one camera at a time, you can actually start to learn approximately for that specific location how to estimate size. People just start to do this intuitively. And so that's something we're interested in bringing in with machine learning down the line, is starting to estimate more of the structure of an individual scene and try to use representations of size then that are more like specific to that area, because you can't actually just use the bounding box size because the animal can come forward or further away or something to represent the size of the animal. Anyway, I'm super proud of all of you. I thought you were really wonderful to work with. And I'm glad you had a good time. I know that, yeah, this is like one aspect of machine learning research, which like I feel like every person who interacts with it ends up doing some data labeling at one point. Actually, I mean, 5th-year grad student, but I've been labeling trees for about 2 weeks. So it's, yeah, it is a very interesting part of it. And we really appreciated your help. And I'll be sure to let you all know how the labels improve the model.
So the idea is once the model is perfected, you could apply this to many other things. You're just sort of using the animals, not so much to study the animals, but to just train the AI basically, right? Or
Yeah,
I
yeah.
mean,
So
you're
I
collecting a lot
collaborate.
of animal data too, I guess.
Yeah, so I collaborate really directly with a lot of ecologists. And part of like this whole area of work, trying to use computer vision for biodiversity monitoring, is trying to help ecologists scale up. So the cameras are actually pretty cheap these days, comparatively. But really, the reason that people can't do like large scale long-term monitoring studies is they just can't keep up with like the data labeling. But if they were able to do so quickly and efficiently, then they could, you could monitor the populations in these areas really, really well. And we can get good models for fixed networks of cameras with a lot of labels. But what we're looking at here is, how do you, with minimal labels, adapt a machine learning model to work better in a new part of the world?
Very cool. Any other questions for either group, or any parting words of wisdom for anybody or anything? Well, thank you all. This has been wonderful. What a fun afternoon of hearing what both groups are working on and what you accomplished. And, very different, but in some ways connected in terms of searching and researching and practicing your scientific method of examining things and looking for things you don't know and finding surprises along the way. So it's fun to hear these two groups present to each other. So thank you all. Thanks to the mentors, the faculty members who supported all of this, the teachers and everybody and students. So thank you all, and have a good school year. It's right around the corner for some of you. All right, bye-bye.
Thank you.
Thank you.
Thank
Thank
you.
you.